Hello, everyone, and welcome to our next edition of Chronicles. We're here with my main man, Mike, that you know, and our special guest, Ryan Lee, Maestro. Despite the fact that we only met a couple years ago, Ryan. Hello, everyone. And yes, Jack, everyone out there, please give us a sign of life that you can hear and see us. It's very crucial. But yeah, Ryan, you might not know, but for myself and a lot of my colleagues in Mexico, Spain, Mike, you've been a mentor to all of us despite not even knowing us, brother. Everything you've written, all of the videos that you've put up in the past have been, at least for me, foundational in changing major perspectives that I had that were incorrect, no other word for it, completely based on science.
And I had the opportunity to sit beside you during the Collectors Cup last year, during CannaReps Collectors Cup, and I have to say, I mean, I was extremely impressed with your approach. You've got such detailed knowledge of varietals, of cultivars, of origin stories of cultivars, as well as the scientific application that you clearly embody. Like, you are a true scientist, right? And I have to say that I really appreciated that opportunity.
Brother,
I appreciate that, guys. Nothing but kind words. Thank you very much.
So today, everyone, we are, what do we have here to sample really quickly?
We're smoking on something that we've been working on for the last year and a half to 2 years. And this here is we're calling it the carrot cake, because you can't not taste carrot cake from this. It is just it is phenomenal.
People who do not smoke smell this flower, and the first word that comes out of their mouth is carrot cake. Doesn't matter, even if it's blind. So it really reeks of carrot cake. It really renders that way on the palate as well. We're going to be tasting this as we start diving into our subject matter for the evening. I have a list of questions that I'm dying to ask Ryan about the origins of the modern CBD movement. Ryan is one of the people that was there in the flesh, that was involved, and I'm just dying to get these questions out. So we're going to get right into the questions, Ryan, if you don't mind.
Yeah, let's jump in.
Awesome. We're going to start with a really brief intro. Just tell us a little bit about your training and the work that you've done, what type of academic training you have. Yeah?
Sure. Okay. Well, I attended university in Ottawa. I did a couple of university programs throughout my time. The first one was an undergrad in neuroscience, kind of a biopsychology thing. So, during that time, I really, you know how it is. You're assigned projects. You have to go off and investigate something of your choosing. And for me, it was always cannabis. So I really, I used university as a time to just research what I was interested in. And it happened to be just a few years after the endocannabinoid system really started being characterized. It was a couple of years after, I think 98 CB1 was discovered, and or anandamide. I'm not sure. I can't remember which first. I think anandamide was first, and then the CBs, the cannabinoid receptors, came in the early 90s. And so I was in university about mid 90s starting off. So it was a pretty young field back then, and we didn't know a lot of things. But it was a great time to learn about it. And during that time, I was already growing cannabis, and it was my hobby plant. I wasn't really much of a drinker back then, and so that was really where I wanted to focus my energies and my attentions as I graduated. So rather than going into lab work on humans or mice or animals, I switched. I took a pretty hard left turn and went to enroll in a plant program at UBC, where I studied like plant genetics and plant breeding and a lot of stuff along that field, biotechnology. So it was probably a little bit early for this kind of thing. Cannabis it was starting to be accepted. I think we had just had the first few medical growers had been implemented, just home patient grows.
Yeah.
And anyway, that kind of just shaped my own development. And when I left university, I was, it was in the seed business, doing work in Europe and elsewhere. And I always attended these scientific conferences. And so at a scientific conference in Germany, I met this American group, and you know, they knew of my work and recognized that I had some understanding of plants, and you know, ended up getting me a visa to go down and work in the States. And we did some massive characterization of the markets down in, you know, California and some of these other legal markets in the States, because we built a very high throughput, you know, sophisticated laboratory for analyzing both cannabinoids and terpenes and other molecules in the plant. And so just throughout those kind of three things, I really got a pretty good grasp of what type of chemistries exist in the plant, on the market, what's not on the market, where are the holes in the marketplace, what kind of things could we make that don't exist yet. And yeah, we just started knocking things off and breeding that way. So, you know, that experience with the laboratory and being able to grow hundreds of plants and send every single bud, or not every single bud, but every single bud from every plant to the lab is really, really informative. You learn a lot of things that may seem counterintuitive about the plant. That's one of the weaknesses of the way that a lot of the research has been done in cannabis is people go to the market and they collect samples, but really the market is just like this tiny little segment of the pie of what actually exists in the species, because you know, it's really based on market preferences and grower preferences and often customer preferences. But there's a lot of things in chemistry, especially when you're looking for specific chemistries. There's things on the marketplace that just, or that aren't on the marketplace that we can create, right?
That's so interesting that you go there, because I mean, we're going to be recurring over that subject matter over and over. And I know also, Ryan, that you're pretty obsessed with the endocannabinoid system, just from interacting with you and reading your posts online. Can you like briefly tell us in your own words, in as few words as possible, why CBD assimilation is so different than THC assimilation in our body? You know, we teach this in our course, but coming from a person like yourself with so much research background, I love the way you put words to stuff, man. You know,
it's a different molecule. I mean, they're very close. People, I don't think a lot of people maybe don't realize CBD and THC actually have the same numbers of carbons and hydrogens and oxygen molecules as each other. They both have the same chemical formula, albeit a slightly different chemical structure. So it's kind of hard to describe in words, but if someone were to go and look up, you know, the chemical structure of THC on the internet or CBD, and you compare the two, they're kind of these three ring molecules with a little tail on them.
Yes.
And that the ring that kind of joins the other two ring, and call it the secondary ring that joins it's kind of in the middle and joins the other two. At the bottom left corner of that in THC, the ring is actually closed. But in CBD, it's a slightly open, the ring is not fully closed. There's kind of a kink in the ring, or a breach in the ring. And that breach is really what's responsible for how the body assimilates it, how the body, you know, which enzymes attack it to, you know, because obviously, you know, in our body, we have these filter systems that are essentially filtering your blood and your, you know, everything that you intake, your whatever water or food you intake. And we identify molecules that are, you know, not supposed to be there or foreign. And then your body has these systems to take those things out of your body and put them into your waste stream. And so the difference in the double bond structure on THC and CBD leads to those things being broken down into different molecules and thus cleared differently in the system. So I think the important thing to take away is that although they're very, very similar, they are actually different molecules. And that's, you know, just because of those differences, the body treats them a little bit differently as it tries to pull them out of the body, right?
Yeah, different molecules, different receptor sites, different sequence of biochemical reactions thereafter, right?
Yeah, well, we don't even really know, I think, everything that CBD does. It's got this broad pharmacology, and it interacts with a bunch of different receptor systems. You know, it doesn't actually bind very well to the cannabinoid receptors. THC does quite well, but CBD doesn't. But it has a lot of these other affinities for opioid receptors and serotonin receptors, and you know, all these other things that exist in the body that are probably beyond the scope of this conversation. But yeah, essentially they're just different molecules, and it's not only, you know, we don't know like I said, CBD is broken down into various molecules. It's one of the ways your body tries to clear these molecules from your system, is it takes it and it breaks it down into these other molecules, and then those things also get pushed out of the body. But it takes time for that to happen. So it could be that some of the effects from CBD aren't a result of the CBD itself, but from things that the CBD is broken down into in your body, right? And they persist for a given amount of time, depending on a whole bunch of, environmental conditions in your body. But yeah, it might be that on the way to those things being pushed out through your waste stream, that they have certain pharmacologies that do good things to your body. So
I guess we're only going to find out more with more research. I heard you say something once, man, that really changed me a lot. You said terpenes are an important part of the biochemical language established between the plant and its environment. So I had this question for you. Do you believe that cannabinoids play similar adaptive/evolutionary roles? And if so, have you observed any patterns in landrace/heirloom populations that in different geographical locations?
Yeah, it's a tough one to say because it's really speculative. I mean, anything that exists in nature at this point in time, I mean, you got to remember life has been evolving for over 2 billion years on the planet. So anything that exists in an organism now, there's a reason for it. It confers some type of evolutionary advantage, and that's why it persists. We also have to remember that the plant that we use today and we see today, the one that's sitting behind you guys and the one that you're smoking right now, is nothing like the plant that humans first encountered. Right? Humans first encountered this tiny little, probably not even a shrubby little plant. It was like a little, tiny little stick that made a few seeds and a few resin glands. And through our association with the plant, we essentially humans, we shape plants to our will. I mean, you probably know Teosinte, Adolfo, back down in central Mexico. It's like the wild relative of corn, right? Well, corn didn't exist. Corn is essentially a big beefed up plant compared to what the wild relative is. The wild relative didn't produce these cobs with hundreds of kernels. It produced these tiny little things with like a few kernels on them. And through our selections, we've been able to beef these things up and make them a lot bigger. And so too with cannabis, right? Like the original thing that humans probably found probably had very little terpenes and very little cannabinoids at all. It did have some of them for sure. But kind of going back and putting ourselves in the context of what the plant did back then is difficult because we just don't know. You can't go and find wild cannabis as it existed back then today. It just, because of the way that it breeds, it shoots off this huge amount of pollen, and we bring it into cultivation and we change it for fiber or seed or oil content, our cannabinoid and terpene content. When those plants are then spewing out pollen, they're sending that improved genetic signal back into the wild. And so they call it a weedy crop complex because it essentially it exists in the wild and in under cultivation at the same time, and those two things are constantly influencing each other because of the genetic exchange that goes back in between the two. So what we have now, what you would find as a feral plant or a wild plant is probably nothing like what we first encountered back then in the day.
Yeah, I got one here. So do you think that it would be possible through either a highly directed breeding project, right? Highly selective, highly directed breeding project, or by fluke, to potentially hit on that ancestor somehow, some way?
The ancestral trait.
Yeah, the ancestral trait, right, exactly. That initial little runt.
Yeah, maybe. Theoretically it'd be possible, but it's kind of like having a map. It's like saying we're going to make a map to somewhere that we don't know where it is.
Right. Yeah.
Right. Like if we don't know where we're going, how do we get there?
Gotta use the stars, I guess. Navigation.
But
we'll probably stumble across signals of it. But again, I would argue that what existed back then is probably not what we wanted. Right. What we want is what we have today.
Right, of course.
Right. So, we want to go back, like probably, you're talking about going back to like the cannabis of the 60s and 70s, Oaxacans and the Acapulco Golds and the real Punta Roja and that kind of stuff, right? The Pakalolo from Hawaii. I think, going back to that far would be good, but going back, even 1,000 years, you're probably not going to like what you see.
Yeah, and that's really more so of what I was referring to. I'm sorry, I should have added more context to my question. In terms of observable populations, do you kind of fall in line with what Merlin and Clark observed in terms of biochemical differentiation in just general senses between narrowleaf and broadleaf populations? You know how they say that there's generally a higher occurrence of CBD in broadleaf populations, less so in tropical narrowleaf plants, but these are not laws that are unbreakable by any sense. And that's not how they pose it. Do you kind of agree with that perspective, or?
Yeah, it's funny. I mean, Ethan Russo said it really well at one point in time when we're talking about, the differentiation between these different species. And, Rob and Mark Merlin and Rob Clark, the two that you're referring to that have done this, BLD, NLD, the broadleaf, narrowleaf type segregation. Ethan said there's really two philosophies. There's the when you're doing this kind of categorization of plants, there's really two philosophies. There's the lumpers and then there's the splitters.
Yeah.
Right. They're the people that want to lump things together and the people that want to split things together.
Yes.
And I think it's useful for theoretical conversations. I don't know how useful it is altogether. I mean, to me, cannabis is one species. It all breeds together. It's more likely that the differences that we see in different types of cannabis all over the world is because people brought initial populations to those divergent regions, and subject to the environment and subject to the uses of what people were doing. That's really what dictated the form of cannabis. I mean, for example, what Linnaeus first encountered as cannabis sativa, which incidentally means hemp, not drug type cannabis. That was a fiber plant, right? That was a plant that humans had bred for the length of the fiber because they were using it for string and rope and all that kind of, not string, but rope. So breeding a larger space between the branches led to longer fibers that were stronger in a rope context. So again, it's like the use of the people that were shaping the crop really dictated the shaping. And I think another way to think about it too is, I mean, everybody thinks, so ruderalis, it evolved in Russia. That's probably not true. Like everything came from kind of that central Tibetan plateau region in Asia. And that's the best evidence that we have, and it dispersed from there. So if you think of, a hemp variety or a fiber variety being brought up to northern Russia either through trade or, some kind of means like that, when the crop, if you think about why it ended up with this short photo period like it did, that far north was not. Yeah, anything that was flowering in November or October even wasn't going to be able to leave its genetics for next season. But the plants that did flower very early, they would be able to make seed and leave them in the soil before the spring, and then that new population that had that really, really early trait would essentially supplant whatever was brought to the area. Right. So it's not only like the human use of the crop that dictates the morphology and all the traits of the plant. It's also the environment.
So what you're saying basically, Brian, is that if we took a purple Kush, a Hindu Kush straight from 5,000 meter elevation, and we were to take it down to a tropical region and grow it for several generations and use it in the way that we use it in the tropical region, which is primarily in flower form and not in hashish form, that this would, over generations, alter it. And you're saying that maybe there might be some co-evolutionary occurrence where it might end up narrow leaf after many, many generations, but it did not come in being narrow leaf original. It had a different morphology.
Yeah, well, again, we have to remember the context is if it moves from seed, if it moves as a plant, as a clone.
Oh, obviously, yes.
It doesn't. It's not really able to change. But cannabis is what we call an outcrossing species, and essentially that means that it breeds more like humans than it does tomatoes, right? A tomato can self-fertilize a plant, and so whatever is a type or a set of characteristics in that plant, they're more likely to be represented in the offspring. Whereas, everybody understands with humans, you have to have another species cohort, i.e. someone of the opposite sex, to have offspring with. And therefore, the offspring inherit not only some of your characteristics, but also some of your mate's characteristics. And so what that type of breeding system leads to a condition where the population kind of carries around all these genetic signals from the past as luggage in like hidden forms, right? It's like they're buried in the genome. And then, we think about the morphology of purple kush. I mean, anybody that's grown these kushes indoors knows they're super dense and tight, but they're much more likely to mold because the flowers are more compact and they trap water. And that's not a very good, if you're grown in a jungle, that's not a really evolutionary advantage trait. And so if the plant would make seeds, it might be that the 1 in 1,000 plant that has a much more loose flower morphology or even a narrower leaf. Like, I mean, if you think about, these fat Afghanica leaves, they don't let a lot of air blow through the plant, right? And that's going to stop your evaporation. It's going to increase your water content in your flowers. It's going to vary humidity content, or at least in the flower, and that's going to make it more prone to rot. So yeah, probably over time, or at least time. It's evolutionary time is kind of hard for humans to understand because we live 70 years, but these evolutionary time, I mean, we're talking thousands of years. So over, at least hundreds of years, yeah, you could definitely shift from, the plants that had the narrow, thin leaves that would allow more airflow through the plant, they might have an evolutionary advantage in a jungle environment. And if that was the case, then yeah, those plants would be more likely to those traits, I should say, not the plants, but the traits of those plants would be more likely to be represented in the offspring.
And that's exactly what I find, Ryan. I think that I'm an avid cannabis traveler. When I travel for weed usually since I've been 15 years old. And going around the world, Morocco, Eastern Europe, China, I find that exactly what you're saying is what I experience, that the plants that have the better adaptation capacity for that environment leave the bigger footprint, and that you can see some patterns in specific type of morphologies, for example, in Africa, that are simply better adapted to survive in that environment. Hence, that's the population that you find there. So, in our school, Ryan, we definitely see value in knowing that there are regional variants. Now, whether we choose to impose, because I also, I'm somebody that likes to present the lumper argument and the splitter argument to my students and let them walk away with the full information and let them so that when they hear Afghanica at level 2, they know what you're talking about, regardless of whether they're lumpers or splitters, right? So
right.
I think that that's the important thing, that a lot of people, I don't want them to walk away from a conversation like this thinking that there's no regional variants, because that's not what Ryan is saying, and that's not what Adolfo says either. There are regional variants. The argument that's being had is whether we should classify this as 2 or 3 or 4 different species or a single species, which is an argument that's based on a whole bunch of pre-existing theoretical frameworks that most of us that are lay people are not exposed to, okay?
Right.
I just don't want people to walk away with too many assumptions, because a lot of us don't have the tools to really take the information that's been discussed and walk away and make it applicable. And the last thing that I want all of you the first thing that I want all of you to remember is that Ryan and myself, when we talk about plants with each other and with Mike, I'm gonna say, this kush is demonstrating this plant is demonstrating some characteristics that are characteristically kush. There's such a thing as typicity, but the evolutionary debate is ongoing, and we need a lot more data and a lot more research to settle this to any final form. Although it's important to note, students, that Ryan's perspective is currently the academic consensus, that splitters are not the top of the heap, even though Hillig, which is the publication that we revisit so many times in class, and Merlin and Clark are both splitters. And that's what I think really confuses a lot of people, because they're like, Adolfo relies on these texts so much and he loves them so much. Ryan Do you like those texts? Do you think?
Oh yeah, they're great for sure. I mean, Robin and the progenitors of Carl Hillig's work. I mean, those are good friends of mine. So I definitely appreciate the work.
Yeah.
I think that type of discussion on whether something is Afghanic or equatorial or whatever words you want, or from India or whatever words you want to put to it is a little bit academic, because even on today's market we can look at our different cultivars. Some of you, I'm sure Adolfo is, doesn't use the word strain, but that really those words they mean a type, right? And any type is describable in the characteristics, right? And I think one of the things that a lot of people do is they look at the plant and they say, oh, it's just Afghanica. And it's like, even within Afghanica, there's so many different subtypes. And I think it's really more important to look at any given plant. And, we can define all these different types of plants that exist on the market today, be it like a purple kush or, a haze variant or like a California orange variant, right? Like, it's okay to split, right? It doesn't have to be about splitting or lumping, but I mean, it is okay to split things apart and say, hey, this is a type, let's describe all of the characteristics. And that could be everything from seed size to the length of the petiole, to the width of the stem, to the height of the plant, to the terpene content, to the terpene profile, cannabinoid content. I mean, on and on and on and on and on.
Yeah.
Right. But yeah, I think it's more helpful for students to learn a plant and not just say, oh yeah, these are Afghanica traits or these are indica traits, but let's look at every single trait that we can characterize on the plant and then try to define it.
Exactly. Let's narrow it down beyond the very, very basic classification system that exists, because I mean, Masari Sharif is such a fantastic cultivar to consider in this conversation, because some of the phenotypes that are described by the people living in that region that have the traditional characteristics that cream of Mazar is made from, they take 20 weeks to finish, and they're very tall, and they don't have exactly broad leaf. That's what I tell people. Not all plants in the Kush region have strictly narrow leaf. Chitral Kush is another fantastic example to look at, where it's not even usually a type 1. The plant grown, if you take the Chitral hash and you test it here, you're going to find spikes of CBD all over the place. So it's about considering every regional variant in its own weights and knowing that, yeah, there's patterns. But, Ryan, this conversation is so great because you add so much color, and that's what it's all about in teaching. Everybody wants to give easy answers, one-dimensional answer. Everybody gets it nice and easy. Listen, this subject matter, if somebody told it to you in a way that's very short and you think you understand it fully, trust me, you do not understand it fully. This is something that takes, I've read about this since I was 14 years old, and I'm still learning. Every time I talk to somebody like Ryan or even Mike has taught me so much, I think it's all of us looking at the evidence together, looking at our plants together, and trying to make sense of it as well as we can, but knowing that it takes actual growing, actual gathering of genetics, actual field work, not just armchair academia, which is what most people discussing this subject are going to bring you.
Yeah, that's really true.
Yeah, man. Yeah, no, that's why we don't know how big the smile on our faces is right now, man, because it's like medicine to have you here talking to people, and to our students and our friends. It's not just our students out there. So I'm going to move on to some more questions because we got some more time, but there's been a couple of interesting questions posed by students as well. So we're going to stop and tackle some as well. Henry was asking, let's go to Henry's question first. Henry was asking, are any consistent rules when it comes to what terroir will affect when it comes to cannabis? That's kind of the question I was asking before. Terroir is being used, just so you know, Ryan, in the French sense, not in the North American sense. Terroir is being used to describe the ecology and the environment in a single terminology, okay? Yeah. So environment and terminology, as Ryan discussed, I don't know if you want to add more, Ryan, but we basically discussed with some detail that, yes, the ecology and the environment, so humans are part of the ecology, right? And those things are humongously influential as to how that plant will look a few generations down. And remember that Ryan specified not just clones. This is the problem with people here in Canada, okay? They're like, well, I'm acclimating my plant by exposing my clone its entire lifetime. That's one lifetime. You have to let it have an offspring in order to really allow the epigenetic changes to take root. If you're not crossing, if you're not actually making genetic changes, right? And obviously there's other ways of making genetic changes today, but short answer is yes, environment, ecology has an enormous impact on how the plant becomes shaped. And yes, there's absolutely some possibility for co-evolutionary occurrence where a plant may not have come from a narrow leaf population and over generations adapted somewhat to have those characteristics to have it be better suited to its environment. Okay, hope that answers the question. And again, Ryan, feel free to.
I'm gonna actually add on to that really quickly.
Please go ahead.
Yeah, so there is a part of it too. That there's a second part of that question, and it says, or does each unique terroir affect aspects of the plant differently without rhyme or reason? I want to say something. I want to speak to what my understanding of not necessarily specific terpenes that are created as a result of chemical constituents within the soil, right? But me being a living soil cultivator, I'm obsessed with the organics, and I'm obsessed with, being able to obviously express the terpenes and the cannabinoids and everything with this plant in the best possible way. And I found that a soil base that has a higher sulfur content in the sense, so like for example, calcium sulfate, right? I found that sulfur helps with terpene production. And I want to know, Ryan, have you heard anything like that as well? So specifically adding gypsum, a higher level of gypsum to your soils.
Your mix, eh?
Yeah.
I don't know about that. I mean, sulfur is definitely a micronutrient in the plant. I don't know how it's because sulfur is typically not a component of terpenes, right?
No,
terpenes are hydrocarbons, but there's also the effect. I mean, the terpene synthase, for example, they need a cofactor of magnesium or manganese to function properly. So, if you have a plant that's deficient in those compounds, the terpene synthases aren't going to be able to work to their peak efficiency. So it could be something to do with that. I'm not exactly sure what the specific mechanism would be. I should have turned my phone off. Sorry. Yeah. So I don't know about sulfur specifically, but definitely, maintaining a complete soil, like a true healthy soil with all the micronutrients that you need, is definitely important. Terroir really means, the flavor. It came from wine, and it really means the flavor of a region. But really, the word really means the flavor imparted to the wine from the local earth, right? So, for example, if you're growing on one side of the Rhone River versus the other, the chemical constituents of the soils are slightly different. So they would say that the wine tasted different as a result of that. And part of that is due to the soil environment, but there's also, as you gentlemen were saying, the environment in a given year in terms of, say, the weather or the air. Cannabis growers, we kind of mess with this a little bit because we mess with it a lot. We don't grow cannabis typically in soil. They do for hemp, but for drug cannabis, people typically build raised beds or they create their own soil. And even if you're in the same environment, the guy on the property next door is probably doing it in a totally different way. So we've got some ways to go before we really understand what terroir is and what it brings to cannabis, the flavors of the plant.
Yeah, these are the research models that are going to be of high interest, I think, to the future, is how different mediums, feed systems, environmentals play into that. I think it's going to be really interesting coming up. We definitely don't have enough data to add a lot of detail to that right now.
It's definitely something that we're obsessed with,
yeah,
and that we think about constantly, and that we're working towards developing phenomenal soil properties. So.
And beneficial fungi and how all of the microbials in that soil also help that all of the interaction with everything that root system is going to see.
Acids, fulvic acids, all of these things.
One little data point might be interesting to your students is that we did a lot of work on this down in California, and we had these very controllable grow environments that were repeat. We were able to run lots of repeats through them the way that the grows were designed. And so we did some growing with, the same system with chemical fertilizers and some with organic fertilizers, and then both conditions with or without a biostimulant. And we definitely found that when you were growing on a chemical fertilizer, your cannabinoids were higher. But if you're using an organic fertilizer, your cannabinoids were a little bit lower, but your terpene content was much higher, right? So there's definitely something going on, and it really, unfortunately, our market is so driven towards wanting these THC numbers because of the way the distribution is done, and the retail customers now believe that, the higher THC content, the better, which is really insane because, when I grew up and we were going to buy weed, we didn't have obviously storefronts. You couldn't walk in and read a lab report or, understand the THC content. The way that you figured out which one you wanted to buy was you stuck your nose in the bag, right? And the one that smelled the best usually was the best.
That's still part of the process here.
So the lab testing in terms of cannabinoid content, I feel, has done actually a real disservice to the marketplace because it's placed this huge emphasis on THC content, whereas I don't feel that that's the most important characteristic for, what I would consider an enjoyable experience, right? It's part of the picture, but it's definitely not the entire picture. Exactly. And you can have, flowers that are 15 to 20% or even slightly less than that and feel perfectly satisfied after consuming them if they have the right terpene profile.
Exactly.
Right. So
Maui Wowie is a perfect example for me, and a lemon Thai, like those two right there never really seem to exceed more than 15%, generally speaking, yet both of those give me such a phenomenal experience insofar as like a narrow leaf goes, right?
Yeah.
Absolutely. Well, I really want to get into.
Yeah, let's talk CBD.
We're going to get back into everyone's questions a little bit. I really want to talk about the CBD research and breeding scene during its earliest phases. I've always wanted to know this. We all know that Musqueam first identified CBD in the late 60s.
He actually didn't.
Oh, no.
No, there was a researcher named Adams in the States. I think he isolated it in like 1940.
What?
Isolated from hemp, yeah.
So it wasn't 68, eh?
Roger Adams.
No, it was well before. It was like 1940. I think his name's, I think it was Roger Adams. But look it up. Look up Adams cannabidiol.
Okay, that's awesome. Because
he also did work with THC in like the 20s. He was doing work with THC, right?
No, they didn't. Well, THC wasn't isolated until in the 60s.
Yeah, he made it
late 60s.
Yeah, 68. Okay, so he isolates Musqueam isolates in 68. So that's where my mistake happens. You're saying that it's first identified in the 40s, eh?
No, cannabidiol was identified in the 40s. THC was identified later on in the 60s.
Gotcha.
And it was actually Guyoni that worked in Musqueam's lab.
Okay.
And I think that they had actually identified the molecule about a year and a half or so before Musqueam finally did. But Musqueam actually nailed down the chemical structure of it.
Gotcha. Okay. So now we know when we identify the particle. When did high CBD producing cultivars start making an appearance? Tell us the story.
So I would split that into two sides. I split it on the pharmaceutical side because GW and Hortifarm were a little bit ahead of down the race than anybody else was. Okay. And then the Spanish folks kind of came along a few years later and found it and then started breeding with CBD. And that's an interesting story. But I think let's start with the CBD that was actually developed by Hortifarm and later licensed by GW Pharmaceuticals. And that was derived from, I believe it was a Turkish landrace and another hemp cultivar that they were using. And they bred the two of those things together and came up with a fairly stable CBD type, which was just, we know now as a type 3 plant. But I think it was only in the 5 to 8% THC or CBD range, very low THC, and one of the conditions was that it could be grown in England because at that point in time GW thought that they were going to grow there. Obviously, if you're a drug production company and you've got a license from one government, it's easiest to do all that work in that one country. When you start having to grow the drug in another country and bring it in, you add complicating layers of permissions and authorizations that you'd have to get from the government. And actually that's what they do now. Or, I think what they do now is they've actually moved it back to England and they grow indoors in a greenhouse. But for a while they were doing all the bulk production of CBD for GW products in Portugal. But like I said, originally they were growing it as hemp in England.
What year is this? Sorry, Ryan.
Okay, so we're talking
90s,
early 2000s. Well, no, Hortifarm was doing the work in Holland on the development of those varieties in the 90s. Yeah, and then in the early 2000s, when GW got permission from the Home Office in the UK to start growing, then they started moving over the production into the UK, and they were, like I said, growing it outside as a hemp crop. But the UK is not an incredible environment for growing cannabis. It's quite humid, it's quite wet, because it's on an island, and it gets cold and wet early in September, and these hemp varieties weren't necessarily bred to be finished then. So they had their first few years, they had a lot of problems with mold, and I think that was why they ended up moving the production down to Portugal. And then later, GW made a contract with this company that produces, like 2/3 of the UK's sugar beet for all the UK sugar needs. And in that process of taking and boiling down your sugar beets to extract the sugar, it's a really energy intensive process, and they were dumping all this heat out into the environment, right? That was just going to waste. And so GW approached them with a project to build a 40-acre greenhouse right beside their sugar extraction refinery, so that all the heat that was coming off the sugar refinery could be fed in to heat the greenhouses. And which is, that's brilliant. Like, that's the kind of thing that we need to be doing in agriculture, because it doesn't make sense to be just, burning more fossil fuels to produce, more fossil fuels to produce heat. So,
yeah. Okay, so that's the pharma side.
So that's the GW side, and that was, like I said, like probably mid to late 80s or early 90s to say the beginning of the 2000s. I think it was around 2008 maybe, or 2009 that they got the CBD hit, and that was from our friend Jaime. I don't know if you know Jaime Garcia from Resin Seeds.
I don't know him, but I've heard of him. Yeah,
a really nice guy, super nice guy. And what they at Squamish, you've been to these events.
Yeah.
There's a nutrient company called Canna, and Canna had put a lot of money into their advertising. And one of the things that they were doing, they had a great booth at Squamish. It was one of the largest ones there, and they had microscopes so that you could come in and see what mites look like underneath attacking leaves. And they did a lot of really neat little kind of demonstrations like that. But they also brought in a gas chromatograph. And so you could show up with a bud and give them a sample, and the next day you could come back and they would have run the sample through the lab. And Jaime had brought in this sample that he had, a plant that he had selected, and it came back as a 7-7 type 2. So it was essentially 7% THC and 7% CBD. And nobody had they'd never seen CBD at that point in time through this particular lab setup. And so everybody kind of lost their minds, or, well, quietly, a few people lost their minds. Wow, we've got a plant that produces CBD. What an incredible thing. And they went on and bred that plant to itself. So I don't know if you guys discuss Etienne de Meyer's Inheritance of Chemical Phenotype in Cannabis paper from 2003 in your class. No. But in that paper, he essentially shows how CBD is it acts like an alternate allele to THC, meaning that if you have one copy of the THC gene and one copy of the CBD gene, you have a type 2 plant. We've refined the model, and we understand now that it's not actually quite so simple, but it does, from a Mendelian breeding perspective, it does segregate in that way. So when they bred that plant to itself, what happens is you end up with 50% of the plants that are, again, type 2, and then the other 50% is half all THC, and the other 50% is half all CBD, right? And so they had to go through a process of figuring out that that's how it worked. And that was kind of when I started working along with Jaime, or at least, consulting with them a little bit. And that was the development of what became the CBD crew, which is now, kind of run under Shanti Bob and Mr. Nice. But it was really developed with Jaime and a couple other gentlemen, Stefan Meyer and Chevy Nadal, that really did the analytics and isolated the plants to find those type 3 plants that they could then breed back with the type 1s to make the CBD 1-to-1s that the CBD crew now sells.
Ryan, I've got a quick question here about this. So there's a bit of bro science out there in the cannabis community about that, and I've heard there's some people actually who I respect as well, that you'll never be able to, and this kind of maybe goes back to that 2003 paper that you're talking about, but you can't ever get more than a 40% total cannabinoid content amongst those two, THC and CBD together. So what I'm trying to say is you'd never see something that's like 20% THC and 21% CBD. That's what I'm hearing, but do you have anything to say about that with respect to?
Lab analysis is a funny thing because there's all these ways to manipulate it, right? Like you get to choose which sample gets to sent off to the lab. There's no, nobody polices what you do to the sample before it goes to the lab. Then when it does arrive to the lab, there's all these mistakes that the labs can make, either intentionally or unintentionally. So where that maximum THC concentration actually lies is probably up in the air. I think it's highly unlikely that you're going to see plants that are truly, truly above 40. You got to remember too, when we kind of do this stupid thing where we have this measurement in the Canadian legal system that we use as total THC. Right? Well, total THC is a theoretical number, right? Yeah. It's a number that's calculated. It's not a number that's measured. What we do measure is THC acid or total cannabinoid acid content. And it may be that plants can get up to 35, maybe 38% total cannabinoid acid content. But even that's really, really high. I mean, you got to think about it. Yeah. One molecule making up 40% of the weight of a flower is pretty exceptional in any plant species. I mean, it's really quite unheard of.
So at the lab that I was working at, the way that we would cross-check results, obviously we would do a bit of HPLC and mass spec cross-reference, but working with the extraction department, we determined that none of the tests were likely hitting above a 34 percentile, really, speaking in terms of the converted particle. Because you can check the weight off of your efficiency parameters, and if the weight's not there, which is, again, a measurable outcome, right, then it probably wasn't there to begin with in your lab test. But yeah.
You got to remember, people got to understand too, this is not like a toaster. Like you don't stick a bud into a machine and then press a button and then 30 seconds later you get an answer.
Yeah.
Right. There's many, many steps in isolating in putting the plant material into an alcohol extract or some kind of solvent extract, and you remove the cannabinoids, and then you have to, if you were to just take that extract and slam it into a GC, it would what we call blowing out the column. You'd have way too much analyte in your injection. So it has to be diluted, right? And every step along the way is an opportunity for error. And so if you don't have somebody that's doing that's designing the extraction method and the analysis protocol, if they're not like the most anal person that you've ever met in your life, they're probably not doing it right.
Yeah.
So just a word of warning, actually, for anyone out there, okay? Anyone out there, Musqueam claims to have 29% CBD and 29% THC.
Impossible.
In a strain called, there's a cultivar called Raspberry Boogie, okay? So it's just to let you know, okay? It's out there. There's a lot of hype about it, but it doesn't exist. The lab tests they're not accurate.
Now,
yeah, and again, let's not, I always try to give the benefit of the doubt. I mean, Musqueam, he's a good guy. I've known him online for many years. I don't think that, he's not running the test. Let's put it that way. He's not doing the lab analysis.
I think it's a typo. It's a typo on his webpage, man, because I think what he meant is it can do 29% THC or 29% CBD. But he put them side by side, which makes it very misleading.
Right.
Yeah.
Yeah, 60% of one compound in a flower. I mean, that's pretty bizarre. I mean, it would look like a glob of Jello, right? It wouldn't look like a flower at that point in time. But yeah, it's always. I think that people just need to take lab results with a grain of salt. And truthfully, if you have like a 25% flower or a 30% flower, and let's just pretend that you actually have those two things, and you roll them up, or two different people take them and roll them up and smoke them, the way that they actually smoke the joint has a much bigger influence on how much THC is going to end up in their system than does the difference between the two varieties, right? Like how long you hold, how deep you inhale, are you smoking in a pipe, are you smoking in a bowl, are you vaping it? Like all these things lead to probably like an 800x it's a much. The point is, it's a much bigger effect than the difference of the actual cannabinoid concentrations of the two buds. So I think that this whole chasing THC numbers that we do is really it's not the metric that we should be using for quality cannabis. If you want high THC content, go smoke flavorless THC diamonds, which I don't consume. I think that they're ridiculous. I mean, they don't have all the good things that I want out of cannabis. I mean, even Rob Clark, who we mentioned before, Clark and Merlin. Rob said, the first time that he smoked pure THC, he said, if THC was all there was to cannabis, I wouldn't be a cannabis consumer.
Straight up, me too.
Yep.
Me too. Yeah, there's other people that would disagree with me, but I am on that boat as well.
It's about the whole rather than some of the parts, right?
Ryan, so the methodology that you taught the Europeans to stabilize the CBD trait, was it a traditional Mendelian method, or was it?
Well, like I said, it was described in that paper, right, in that 2003 paper, Inheritance. And so I kind of showed them the paper, and I was like, "Hey, you guys, this is why you're not getting the uniform results." And I think one of them had actually already figured it out, and they were in the process of experimenting. But I only backed up their experiment. And then our another mutual friend of that group and myself, a guy named Roberto from Grassomatic Seed Company, he was tasked with, because he was really kind of big and not popular, but he was doing a lot of good work in the feminized autoflowering space. I think he was one of the first people to commercialize feminized autoflowering plants. And he wanted to make a CBD 1-to-1 because he had a family member that was had this really serious brain inflammation condition. And it was, she was very, very sick. And at the point where she was like, the doctors were kind of giving up. And he got a hold of Jaime and, started her on the 1-to-1, even just taking decarboxylated flour and putting it into capsules. And she was taking the capsules just as decarboxylated flour. And it did tremendous things like this. Yeah. They wanted to put her on interferon gamma. And immediately when he started taking the THC, the doctors were like, what happened? Like, we don't understand. She was so sick and now she's so okay. And so anyway, Roberto making this autoflowering CBD 1-to-1 was really about finding a way for anybody's grandma or mother to be able to take seeds and plant them in the ground and get a medicine out of the material. So I helped him design a breeding program that would both maintain the autoflowering trait, and then we could create type 3 CBD plants so that we could then create the 1-to-1 hybrids. And that was kind of my involvement. And so it definitely wasn't a key player. I mean, Jaime and Scott from Shanti Baba, Scott Shanti Baba from Mr. Nice Seed Company, and Roberto from Grassomatic. They're really the pioneers. They're the ones that did the work.
It's awesome of you to be like that, Ryan, because I mean, in Spain, you're so well loved, man, that I dropped your, like, honestly, they're like, oh, you live in Vancouver, you know Ryan? I'm like, no, man, I wish I knew Ryan. So it's nice to know you now, man. And
Spain is such a great country, man. I mean, it's so beautiful, and the people are so friendly and nice. And, despite, I mean, they're getting a little bit kicked in the teeth now on the commercial side of the cannabis business, but with the social clubs and the home growing, the toleration of home growing, it's a pretty good model for a country that's still under prohibition. So I got a lot of love for the Spaniards, and that whole
that whole scene,
that whole community. Yeah.
So tell us what you're up to now. Where do you see yourself going for the next little bit? Where's CBD going? Are you still working on CBD projects? Are you
a little bit? The work. So what in the years after that CBD crew thing went down, that was when I really was doing the work in California, and so we made in that time we made CBD plants in every terpene flavor class. So we made. The step is to make type 3 plants. So you take a THC plant with a flavor, you cross it to a CBD plant, and then you inbreed it to the point that you remove all the CBD or the THC plants, and then you have. I should go back. The original CBD plants didn't have a great flavor. They were all myrcene dominant, but they also kind of had what I consider this. I call it wild, like a sweet hemp smell.
So there was an association between myrcene and naturally occurring CBD populations.
Yeah. The reason why is that myrcene dominance is the natural state for all cannabis, in my opinion.
I agree with you 100%. Same conclusion.
Yeah, if you look at all the different hemp varieties and they do these mass distillates of the populations, it's always myrcenes in the top. And if you look at like cannabis's wild relative hops, even in hops, the majority of hops are myrcene dominant as well. And so, over the last 30 years, in us creating these different smelling plants, we've always been selecting away from myrcene. But when we went and started working with the CBD populations, they also were from wild populations, and so they hadn't been pushed towards THC, and they hadn't been pushed towards anything other than myrcene dominance.
What type of regions were they collected from, Ryan?
I'm not actually. That's the one thing that's missing from the Spanish CBD story, is that nobody really knows where there's all this confusion, right? There's some people say
three things already. I've been told 3 things by 3 different groups. I spoke to CBD Botanical, CBD Crew. All of them have a different story. Yeah. It's probably some kind of heirloom that came in from North Africa, but we don't know.
It could have been hemp. I mean, it really could have been hemp. Who knows, right? There is a lot of hemp grown in Europe, so it's quite possible that pollen flies around. So it's possible that the allele was introduced that way. Mm-hmm. And so once we got these populations and started doing really directed crosses, we were trying to boost up the cannabinoid levels, the CBD levels, but also we were trying to create different entourage families where you had CBD in the context of all these different terpene profiles that we've developed on the THC varieties. And so that took a couple of years of work, but it was really enjoyable, right? It was making all these 1-to-1s, and in the process we discovered all these really interesting things. Like we made 8-to-1s, for example, which 8 THC to 1 CBD is a ratio that's never really seen in nature. And it provides just enough CBD that it doesn't really interfere with the high, but it takes a little bit of the edge off.
Yeah.
Phenomenal.
Yeah, that's what I love about Mazar. A lot of the Mazars, a lot of the older heirlooms that you get from Morocco, they have nice little CBD spike. They just feel so different, doesn't it? So different.
Yeah, it changes, and we figured out how we could play with that dial, and there's a whole genetic switch on how you can play with that dial. But that was a real deep dive, and I haven't, other than using CBD varieties for breeding, different propyl plants, strangely enough, CBD is the type 2 flower is not a thing in Canada. And it probably goes back to this thing where everybody's chasing the highest THC level. If you have THC and CBD, they kind of like split the total cannabinoid fraction. So, let's say that there's a limit of 30%. So that's not a real number, but let's just assume that it is for the purpose of this discussion, that there's a limit of 30% total cannabinoids in the plant. One condition is to have all of that being THC, but if you want to add in a little bit of CBD, that dial has to turn down from 10 to like 5 or 6, right? So you have like, rather than having 30% THC, now you have like 15% CBD and 15% CBD or THC. And if people are buying only on the THC metric, that kind of pushes type 2s out of the market.
In Canada, yeah.
And so that's one of the reasons type 2s aren't popular on the market, I think. Another one of the reasons is that all the stuff that ended up on the market in Canada as type 2 plants were myrcene dominant. Mm-hmm. And they had that kind of wild, sweet hemp smell, which is not really, people that are smoking OG and smoking these high terpinolene varieties, anything with a lot of flavor, you go back to something that tastes like hemp, you're not really satisfied with it.
Well, that's why Afghan #1 is not in the market anymore, man.
Right.
It tastes like dirt.
Yeah.
I used to bring that into the classroom to show students. I'd be like, look, this is what we made stuff like cheese from and blueberry, but do you want to smoke the ancestor or do you want to smoke blueberry? You know what I'm saying? Anyways, Ryan, I can't tell you how much enjoyment I derive from today's session, brother. It's gracias from the bottom of my heart. We're going to do our giveaway really quickly. Just so you know, Ryan, every time we give away something at the end of our session. Today, we're giving away a free tuition. I'm also, of course, going to be giving all of you the academic publications that are related to today's conversation. So I'm going to put, Sharon is going to put those on the screen right now for you to download. You're going to get the full text of Merlin and Clark, of course, and you're going to get the full text of Hillig, of Carl Hillig. And I will be sending you in an email the 2003 publication that was referred to by Ryan, because I've read it and it does need to be included in my class. I actually only read it a few months ago, Ryan, when you mentioned it on Twitter. So I wasn't exposed to it that long ago. It was a really eye-opening experience to me, and I'm so stoked to share that with my students. So on your screen right now, you've got the academic publications for download. The next thing that we're going to do is Sharon is going to announce the winner of today's draw, where we're giving away a free tuition to our level 1 course. So somebody is getting a free tuition today. Sharon, who will it be? Please, Sharon, let us know. Sharon, just so you know, Ryan is like this angel that hovers above us, and she takes care of absolutely everything that has to do with administration for CannaReps. So we would not exist if it was not for Sharon. Our winner is, who is it? Oh yeah, Jamie Happy. Jamie, you have to answer on the chat box if you're actually going to claim your prize. Remember that only the people who stay for the end of this educational session get their prize. You have to say something. Is Jamie here? Oh, yes. Jamie, no. Jamie has not said anything yet. Don't claim victory just yet. Otherwise, you have to give it away to somebody else.
Yeah.
So,
Really?
Jamie, speak now or forever hold your peace, Jamie.
Got 5 seconds, Jamie.
I don't want to put any pressure, undue pressure on anyone. I'm sure that Jamie has it on the background right now. Oh, no. Is that gone?
That's 5.
Sharon, I think we need to do a new draw. Oh, no. Jamie. Oh,
Jamie
he
went to the washroom.
Okay. Greg has claimed Jamie's prize. Greg, thank you for doing that.
Hopefully Greg knows Jamie.
Yes. I hope that... Wait. Greg, was that a joke? Or did you actually see this person go to the bathroom? I haven't seen any action on here.
Yeah.
No confirmation. Joke. Okay, it was a joke. Sorry, Jamie. Sharon, who else? Who else is in line to win this? Unfortunately, we have to move on. Try to draw from somebody that's not a previous student, Sharon, because of, yeah, sorry, Jamie. I'm sure that Jamie's going to check out the recording and be angry. I'm not stoked about that.
No.
Our next winner is, I'm sure that we're going to hit gold this time.
Yeah.
Strathian.
Cool name.
Strathian. And you know what? Gotta be here because we started with just under 100 people and we still have 63. Chances are very high. Yeah, Strathian got it. Very nice. Congratulations. I look forward to seeing you in class. I'm sorry if I'm totally massacring your name. I have a name that is very commonly massacred as well. I know what it feels like. Strathian, I send you congratulations. Ryan, thank you again, brother. I cannot tell you how much this was a benefit to me, to my students, and to our friends.
Ryan, it was great once again, as always.
Hasta luego, everyone.
Hasta luego.
And we will see you next month. Gracias to everyone.
Cheers.
Bye-bye.